Alright, you should be able to do this whole worksheet on your own without my help, but I'll go over it if you have it. So, chromosomes, remember, are structures that hold DNA in the nucleus. Alright, and then this is just going off of the um, sex-linked genes that we did in the Flintstones. So I'm just going further into that. And the first thing we're going to look at is mutations with chromosomes. So how many chromosomes in our body is, remember, there's 23 pairs or 46 total chromosomes. So if you count all these up, 1, 2, and you just go over all the way across, you count 46. Autosomal chromosomes are all the ones from 1 to 22, and the sex chromosomes are the X and the Y. So these are the sex. All right. So the ones that we're going to concentrate a lot of in this section is going to be the sex chromosomes. Alright, so now we're just doing some quick review here because we're going to talk about mutations. We already talked a little bit about mutations in the DNA, but we're going to go over the chromosomal mutations that you can get. So, meiosis makes 4 cells with half the number of chromosomes, or you can say 23 chromosomes. You can also say these are gametes or sperm and egg, whatever you want to say for that one, and it's with 23 or half the amount of chromosomes. Alright, so males, the sex chromosomes that males can pass down is they can pass down the X or the Y, whereas a female will only pass down the X chromosome. Alright, so that's a key point in when we're going to get to sex-linked traits in a second. Fertilization then occurs between these two, and it will make a zygote or an offspring that will have 46 chromosomes. It gets half, or 23, chromosomes from the mom and 23 or half from the dad. Alright, so we get an equal 46 then. Alright, so now we're going to look at a chromosomal mutation and what happens when we do not get exactly half the chromosomes. So let's look at mutations. So the first, just review, there's 2 types of mutations here. There's mutations of a gene, and then there's mutations within the chromosomes. So remember, a gene mutation is if you have a change in the base, or the DNA, which affects the amino acids or the protein. Alright, remember proteins are made of amino acids, so if it affects the amino acids, it affects the protein. And what is a gene? It's just a segment of DNA that codes for a protein. Alright, so example of gene mutation is if you have like AAT, AAC, and then that gets mutated to AA, AAC. Alright, and that will then code for different amino acids. I don't have the chart in front of me, but let's say this for Lu, and then this is cis. So then let's say this is THR, and then this is cis. So it changed that amino acid, which then will change the protein. Alright, chromosomal mutation down here is a change in the number of chromosomes, which can have an effect also, just like these proteins. Alright, so here's 3 examples here. So the first one is Down syndrome. Down syndrome, if you look through, there's a pair at 1, pair at 2, pair at 3, pair at 4, so on and so forth. If you go all the way down to 21, there's 3 chromosomes. So that means they got, instead of getting half, 1 from mom and 1 from dad, they got maybe 2 from dad and 1 from mom. Alright, so what happened is during meiosis, the chromosomes didn't split. So somehow they got an extra pair of chromosomes. In Klinefelter syndrome here, if you look here, they're all normal pairs except for here. There's a pair and then there's normal one, so they got two X's and a Y. So again, either the mom gave two X's or the dad gave an X and a Y instead of an X or a Y. Alright, so again, the chromosomes did not separate, stayed together during meiosis. Alright, and then still the last one here now is there's only one X. Alright, so instead of having two X's or an X and a Y, you only have one X. And this is also because it didn't separate, but then instead of getting two, so maybe this baby, the baby up at the top here, for Klinefelter syndrome, maybe that baby got the X and the Y, so there was a gamete here that had the X and the Y, and then the other gamete didn't have either. Alright, so then from the mom, it got an X, but then the dad didn't donate anything because when meiosis happened, two went to one set, both of the pairs of chromosomes, so like both the X and the Y went to that cell when it went through meiosis, but there was no X. There was nothing else to give to this one because the dad only had these two chromosomes to give out of that pair, so the other one was left empty. All right, so when then this one combined, when the sperm combined with the egg, which only had an X here, it had all the other 44 chromosomes, but then just one X, instead of having an X and a Y or two X's. Alright, so all these are just due, if you know the basics, just due to a problem in meiosis where the chromosomes did not split apart. Alright, so that whole first part was just to get you remembering about meiosis and fertilization and talking about other problems that can happen. So a lot of this happens due to meiosis, and so like Down syndrome is more common when you're older because the eggs are going through meiosis longer and causes more chromosomes to happen. Alright, so now sex-linked Punnett squares. So you did these a little bit with the Flintstones. So to the left is a Punnett square. And what was the mother's genotype? It was X. Remember, mother's XX. And she was heterozygous, a big H, little h. The father was XY. And he just has a big H. Alright, there is no allele on the Y chromosome. There are only alleles on the X chromosome. And so sex-linked genes are the ones only found in the X chromosomes. So if we do some examples, I'll just do one here, and then you should be able to do the other one. Now the key problem, a female is homozygous dominant. What percentage of the... Actually, I might do two. Let me look at the other ones I have. Alright, I'll do this first one, but I'll talk about all the different products. So, female, so XX, is homozygous dominant for no color blindness, and male is colorblind, so XY, and you have a little h. Alright, so you fill this in. Alright, so both of the females get X big H, X little h, and then both the males would have X big H, Y. Because the male got, since the female, the mom was homozygous dominant, it gave both a X big H to the dad. So what percent of the children would be colorblind? It would be 0%. Alright, all the females here will be carriers. They carry the recessive allele, but don't pass it, but don't have it. And all the males will be normal. Alright, so you guys can do the other ones, and you should be able to answer the questions at the bottom. So once you do all of them, who makes males get the sex-linked trait? Well, remember, sex-linked's only on the X. And since males have an X and a Y, they got the Y from the dad, so they always get their X from their mom. Who has a greater chance? Males have a greater chance because they're either have it or they don't. They can't be a carrier. All right, so only females can be carriers when it's sex-linked because they can have this. Males would only have that. All right, so males are going to be more likely to get the recessive gene. All right, so let's quickly look at the pedigree, and I'll do one or two of these, and then we're done. All right, so you should know all of these questions here, the first one, so I'm going to go down to number 1. Well, actually, I'm going to do number 2. I'm going to show you that example because there's a little bit of trickiness there. So person number 2, it's right here. That's a female who's a carrier because she's halfway filled in. All right, with a colorblind male. All right, so if we do this, we got X, 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 Y, X, Y. All right, big R, little r, big R, little r. Oop, little r, sorry. Yeah, this one's a mess. My dexterity is not good enough on this board. And then X, big R, Y, X, little r, Y. Alright, so this question says, what is the percent chance that a female with colorblind, what is the percentage chance that she will be colorblind? So we're looking at just if they have a daughter. So what you can do is actually cross out the males and just look at the females because we know that they're having a girl. All right, we looked at the ultrasound, we saw that they're having a girl, and we find that this girl here is going to be colorblind. This one's not going to be. So it would be 50%. So instead of being 25% chance, it's a 50% chance because you're only looking at the females. And then here, this one, we'll do this last one here. So person number 3 is another female who has little r's, colorblind with a non-colorblind person, so big R, Y. So these are both going to be X, big R, X, little r. And then X, little r, Y, X, little r, Y. Alright, this time they have a male. It's kind of hard to see there. And what is he going to be colorblind? So now we're only looking at the males, so we can cross out the females. Only look at the males, so both of those chances for have a male would be colorblind, so it would be 100% chance that the male will be colorblind. Alright, so when you read these questions, this one says child, so we can look at any of the children, regardless of the gender. If it says female or male, you have to look at just the females or just the males. Alright, so and then you can ask me questions if you're confused on the other examples that you have to do. And that is it.